today. We're gonna go through a brief description of what the FASEB DataWorks program is here, and then we'll jump into some really engaging talks by two experts in the field of data integration. So DataWorks is a program of FASEB. We're a federation of professional societies who work in experimental biology. And this program is really focused on linking researchers to skills, resources, and materials that help them learn data management and sharing skills. And we have three parts of our program: the prize, we're learning from two of our prize winners today, our salons, where we are today, and then our help desk, which is a great tool that I'll talk about in the end to bring people into learning data management skills and eventually also the best practices. So before we jump into the presentations, I do want to give space for a poll. Again, you can join us on Slido, but I wanted to ask a question. Oh, all right, that didn't work. So we're just going to go into the talks. It's pre-filled a little bit and we can't predict that. So we'll jump into our first speaker, Monica Munoz-Torres, who is an associate professor at the University of Colorado, Anschutz Medical Campus, quite a mouthful there, and the program director for the Monarch Initiative. So I'll stop sharing and turn it over to you, Monica.
Thank you very much. Give me 1 second. I'll start sharing it myself. You should be able to see my slides now. And for those who want to wish to follow along, you can go to my slides. They're always publicly available. Thank you so much for giving me the opportunity today, Emily, and it's been a delightful journey for the Monarch Initiative to be participants and award winners for the prize that was given on data sharing. So today I'm going to talk to you briefly about a little bit about the what and the who is the Monarch Initiative. And so I'll talk about the impact that we believe that we're making, our efforts for community engagement and outreach, and I'll share a little bit of details about our team at the end. So I'll just dive us right in. If you forgive me for 1 second, I'm going to hide my controls. There we go. Well, thank you so much. So the Monarch Initiative, we're an international consortium, and we lead key global standards in semantic data integration technologies. We were launched in 2013, and the initial goal was to leverage all the phenotype data from across organisms and sources so that we could improve rare disease diagnostics. Because for so many rare diseases, there's simply just no known underlying mechanism in humans. And so the Monarch resources in the integrated data are now foundational to many downstream applications and contexts. And Monarch works closely with a variety of communities to help standardize our data for reuse. And so to maximize the utility and the impact, the Monarch platform is composed of multiple open source, open access components. And we promote provenance and transparency in the enhanced use of standards and new technologies. And we also promote improved data accessibility and user utility in data submission. So we are, as we call ourselves, big data, research parasites. But aggregating information and bringing it all together is simply not sufficient. So before I dig into the details, I'd like to share some thoughts that underlie what we do at Monarch. We believe that it is imperative to connect resources in an interoperable manner to make them computationally discoverable by all of you. So in order to view the big picture, detect the gaps, and allow both humans and machines to draw these like cross-domain insight, we believe it necessary to conduct careful alignment of all the distributed data to shared conceptual frameworks and to use consistent identifiers. So the integrated data paint a much more comprehensive picture, as we're trying to represent here, and it allows us to better leverage existing knowledge to address our questions. And so the Monarch Initiative basically enables the possibility of data reuse and the collaboration at scale, and this catalyzes biomedical research. We believe that it does so, and we do this first because we leverage model organism data, which allows us to unify and harness the wide range of data using ontologies and data integration technologies. In this way, we increase the utility for researchers across diseases and anatomical systems and species. And then second, we also galvanize translational research and precision medicine, because we have the most comprehensive open collection of gene to phenotype data in the world, and we would like to believe so. And linking together clinical and biomedical and basic science research data across a wide range of organisms and patients and body systems and diseases enables this. So maximizing the reuse from a broad and disparate landscape of disease and phenotypic knowledge and data sources, Monarch has helped to fill a critical gap in the availability of gene to phenotype data connections. So for, and also, I wanted to say that, for over a decade, we've been creating semantics tools to harmonize the data for computational reuse, and the lack of consistency that exists in how each data source curates different associations, such as like the disease to the variant or the gene to the phenotype, is really challenging. And some of the core strategies we employ to address include the best practices that we've developed for identifier management and reconciliation, and second, community development of species neutral ontologies, such as Uberon and the VEuPheno ontologies. So Monarch data reuse is also notable for its scope and for the mechanism discovery that happens across species and diverse domains, like the rare disease diagnostics, evolutionary biology, veterinary medicine, biodiversity, and this, the wide range. This is wide-ranging impact of the Monarch resources. We believe is because it standardizes the entirety of the anatomy that exists in the gene ontology, and it helps to realize precision medicine through patient classification, and it supports other resources in their efforts to become interoperable with schemas and identifiers and semantics. And with our resources, we assist clinicians and researchers to improve the lives of patients that have rare diseases. So to hone into this integration, by making data more interoperable, our widely used standards for data annotation and exchange help us to support a wider range of data sharing and reuse by projects and organizations around the world. And this reduces the effort that they need to devote to data harmonization. So the VEuPheno ontology is trying to unify the annotation of phenotypes across species. The Mondo and VEuPheno unify existing terminological resources into ontologies that are semantically coherent. And this is a huge advance for the rare diseases community. The Mondo ontology is already the basis for many organizations' additional curation clinical diagnostics, for instance, ClinGen at UNC and many other organizations across the world, the NLM, MedGen, GA4GH, the European Rare Disease Reference Network. So this has multiple effects. This has a multiplier effect, rather, and it brings together multiple experts and stakeholders to work together on how to best categorize and catalog disease. And then we also, of course, have our PhenoPacket standard for sharing diseases and phenotype information, which has been designated as both a Global Alliance for Genomics and Health and an ISO-approved standard. And this is a widely adopted a standard now by many countries. And of course, the Human Phenotype Ontology, which has been made more accessible by being translated into into many, 20 European and African and Asian and Australian Aboriginal languages. And so this is what integration looks like from the perspective of the Monarch ontology. This is the representation of how our knowledge graph is structured. The three primary data types in the Monarch knowledge graph are genes and diseases and phenotypes. And the image here details the entirety, the entity, sorry, so each node and the edges, or the counts and the unifying ontologies by which the source data and the ontologies are harmonized. So this is about the data harmonization that happens within the Monarch Initiative. So cross-species inferences that are demonstrated by these graphs over here are accomplished via the gene orthology, and via homology and phenotype similarity. And then the content dissemination of all of this integration is done via the APIs, the Monarch UI, and in clinical applications as well. So our goal is to galvanize translational research by developing tools like Exomizer and data services like those that we developed for the Anschutz biomedical translator, and analysis approaches that help diverse scientists and perform analytics on genes and phenotypes and related data from a range of sources. And so I'd like to share this story about our work trying to overcome an intractable health challenge, which is the diagnosis of rare diseases. It's incredibly challenging to diagnose rare disease patients, and the diagnostic utility of the whole exome analysis has been, hovering at around 30 to 50%, depending on the papers. And fundamentally, we need more data to make better diagnosis. And Monarch has substantially helped us to improve this situation. So almost all genetic diagnostic tools now use the human phenotype ontology, and we've been able to demonstrate that there is increased efficacy in a number of settings. So, for example, the 100,000 Genomes Project, which has genomes from patients with rare diseases or cancer that were sequenced to find a diagnosis, had a diagnostic pipeline that would utilizing Monarch's Exomizer tool, which is it's a variant prioritization tool. And this leverages the genomic and the phenomic information in this ontology-driven phenotype matching algorithm can identify patients who have an atypical profile for a disease and can help solve some tough cases. In the study that's described in the paper that's referenced there, here in the link, Exomizer actually found that 80% of the rare diseases diagnoses were being listed in the top 5 ranked candidates. So give it a read. It's an exciting way. I also wanted to share with you, Emily was asking questions about, like what's next. So we're advancing Monarch tools and methods using LLMs. And as a matter of fact, I saw a few of our incredible developers that joined the call as well. So over the last 6 months or so, we've been partially pivoting and rethinking our strategy in terms of large language models, for instance, using GPT-4, and we're actively exploring opportunities to use them throughout the Monarch stack. So we've been using text-based generative AI, the Monarch Assistant, which is a Monarch plugin of the OpenAI ChatGPT that actually uses the curated knowledge that we have in our knowledge graph. There's the Talisman, which basically uses large language models to interpret lists of genes. There's our work on Spires, led by Harry Caulfield, on how we parse text to knowledge graphs, finding evidence in the literature. This is a work in progress called Curate GPT, and finally mapping ontology terms using Mapper GPT. So if you went to my slides, you can see all of those links, and you can also learn more on our GitHub repository for OntoGPT. So finally, I'd like to tell you very briefly about our efforts to bringing the community together around our resources. So first, we truly believe that Monarch serves as we call it a socio-technical model for the creation of community-driven, fit-for-purpose standards. And these resources we provide includes cross-domain open standards and libraries that have required years of development and also years of community building. We'd like to say that if Monarch weren't there, then many projects in biodata resources would have to have reinventing these tools in these standards. It doesn't mean that none of it would exist, it means that some of these processes would go a lot slower. So our resources are used across the biodata resource ecosystem, and it includes some of the largest open science efforts that have been funded by the NIH, like the Anschutz Biomedical Translator, the Center for Cancer Data Harmonization, the National COVID Cohort Collaborative. And these projects would also have to socialize their own standards in the absence of Monarch. There are resources such as Exomizer and Mondo and HBO and then PhenoPackets that I mentioned that have helped to democratize diagnosis, and particularly for rare diseases. And this perhaps can offer some hope for researchers and clinicians and the patients themselves. And HBO's support for a lay person terminology also enables patients to communicate their symptoms and to clearly state to their healthcare providers what they are feeling. And this actually gives them the possibility of taking a more active role in their own medical journeys. And our PhenoPacket standard finally is increasingly used by tools that export patient profiles, including phenotypes in the patient archive in Australia, and the IERUD exchange, and the European Joint Program for Rare Diseases. So these are just some examples on how HBO, which is in use across the globe, is helping demonstrably is helping to improve diagnosis for rare diseases. So I'll let you go with a thought about how all of this depends on finding consensus across the community, right? Like, I'm pretty sure that in our field, community developed and open standards are critical, but knowing which standard to use and by which criteria to employ it and what actually constitutes a standard is a little challenging. And there's no such thing as, a recipe that connects the domain to the data standards that they follow. So the right standard to use really depends on the use case that you have for your given project. In some cases, the potential downstream applications for the data that you're aggregating or utilizing or generating may not always be apparent as technology evolves. So finally, just, the communities and individual adopters have to strike a balance between planning for the future and minimizing the burden of compliance. And I'm here to tell you that, all of the tools that we use employ semantics as a layer that we understand to be, the universal converter. So all of these are resources of the Monarch Initiative, the Mondo Disease Ontology, Human Phenotype Ontology that I shared, the LinkML framework in the BioLink, the COSA ingest, the mapping SSSOM, PhenoPackets, the the treatment medical actions ontology, environmental ontology, Uber for anatomy, and Euphena for anatomy. I would like to thank you for your attention and share with you the team that participates in the Monarch activities. These are our members. Some of them are here, and thank you so much for joining us. And I am where I am and can look high because I stand on the shoulders of giants. So these are the PIs who lead the products and resources development for the Monarch Initiative. So Melissa, Chris, Peter, who's here actually, Damien, and Jace McLaughlin. Thank you so much. Ian, the slides are in the chat, so I'll be delighted to let the talks continue, and then we'll go into our panel.
Great. Yeah, there's so much you covered there. And in particular, I hope to ask you during the panel more about semantics, because I think you're very skilled in that, and there's a lot that we can learn in that realm. Now we'll turn it over to Omar Harb. He's the Director of Scientific Outreach and Education at University of Penn's VEuPathDB.org project, and I'm excited to hear more about this work, which was also an awardee of the 2022 DataWorks Prize.
Great. Thank you, Emily. Hopefully people can hear me and you can see my slides. Okay,
Yes.
great. Perfect. All right, so I'm going to walk you through. I have a lot of slides, so I'm going to go through them very quickly, but I can also skip a bunch of them as needed. So I'm going to tell you about VEuPathDB. VEuPathDB is one of 2 bioinformatic resource centers. This program actually started very early in the 2000s. So the first award was given in 2004, and subsequently every 5 years, the contract was competitively, awarded to many different centers. The awards have decreased in number over the years, and as you can see here on the bottom, the increase, as we all know, the increase in the number of the amount of data, in particular here focusing on genomes or data in GenBank, has continued to increase very rapidly. But the number of these centers has reduced. Not necessarily a bad thing. I mean, things get more efficient, compute power is better, and things get more automated. So this is maybe a natural the way things should run. We are at the final year of our last contract, and we submitted a grant for the next round. So hopefully, if we're lucky, we'll be back with you next year to continue our efforts. As I mentioned, VEuPathDB is one of 2 bioinformatic resource centers. These are funded by the National Institute of Infectious Diseases, Allergy and Infectious Diseases. One of them is BVBRC, which supports bacterial and viral pathogens. And if you're interested in bacteria and viruses, I would encourage you to go to bvbrc.org. They have a lot of incredible tools and very useful features that may benefit you. VEuPathDB, which I'll tell you about today, supports eukaryotic and fungal pathogens. Mostly protozoan pathogens and many fungal pathogens, obviously. Their hosts, so humans and mice, and also the invertebrate vectors that transmit those diseases. So things like mosquitoes and ticks and sandflies. This is a multi-center project. The key PIs on the project are Marianne McDowell, who's based at the University of Notre Dame in Indiana, and David Roos, based at the University of Pennsylvania, where I am based. And then a number of other lead investigators based in different centers around the world. I would like to point out in particular the European Bioinformatics Institute, where a lot of our data actually gets processed, and we work synergistically with them to transfer data back and forth and make sure everything is harmonized and working together. As I mentioned, the bulk of our funding comes from NIAID, but we also have funding from the Wellcome Trust to support some of the projects in the UK. In addition, we've received funding from the Bill and Melinda Gates Foundation in the past for a couple of projects that I have time. I'll mention them in the end. Our philosophy really is to, in the end, is to give the researcher in the lab the tools and the ability to interrogate the huge amounts of data that's been collecting and accumulating both in repositories and in publications over the last 20 or 30 years. And what's important, obviously, is allowing people to ask those questions in a way that's easy for them to do so and doesn't require an extra degree or a whole new set of skills in order to ask those questions. People are busy anyway in the lab, and we want to make sure that the data is accessible. This model has been successful. So as I mentioned, we've been around for close to 20 years. This is showing you just our Google Scholar citations. We're up to almost 2,500 citations annually for one of our for our resources. Sorry, I jumped over. And another important thing to point out is that we've recently been recognized as one of the global core biodata resources. So indicating the important position of VEuPathDB, but really not only VEuPathDB, but resources like VEuPathDB that whose goal is really to make data accessible, interoperable, and available for people to use. We're a true production resource, so most of the people who work on the project are actual staff and employees. Very few postdocs or actually no graduate students, very few postdocs. This is really based on providing a service to our end user. We have releases once every 2 months, and our releases is our release process. So our data integration process has multiple steps. I won't go through the details here. Suffice it to say that we have standard operating procedures, and for every release, we go through these procedures to from anything from identifying data sets to prioritization to making sure the data is processed in the same way and that the data is QC'd. One thing to point out is that we rely very heavily on our community to make sure that the data that is in our resources is accurate. And so every data set that we integrate, we interact with the people who generated the data, the authors of the publication, or if it's pre-publication, the people who provided the data, and we ask them to provide a certain level of QC of the data before we release it publicly to on the websites. Pointing out here that all of our data sets go through standard pipelines. And so again, we're not going to go through the details of this, but the bottom line is that we have a lot of standardized workflows where all of our data gets processed through those same workflows so that they can be compared in certain ways between each other. And in addition, all of these get integrated into databases, and we provide a number of services that are accessible by our users in order to interrogate the data and use it. And more importantly, this little pink part here on the top is the user interface, or the web interface that most of us would interact with. And our goal, we focus a lot on making sure that this interface is user-friendly and allows users to actually come up with novel ideas from interrogating the underlying data. In terms of the types of data that we integrate, this is a list of some of them, just highlighting some of the key ones. I mean, any transcriptomic data set, proteomics data set, post-translational modifications, metabolomics, cellular imaging, phylogenomics. So the data this data comes from either from known repositories like the Sequence Read Archive or PRIDE for proteomics, but also from providers directly. Many data sets do not have their orphan data sets, they don't have repositories, but still we integrate these as they're incredibly valuable for our users to be able to ask questions and come up with some useful conclusions that they can test in the lab. So I'm going to walk you through some of the tools that VEuPathDB offers that have been successful in allowing users to come up with new hypotheses, importantly, new testable hypotheses. The first tool I'm going to walk you through is the search strategy system. So, and this system really allows you to build an in silico experiment. And so looking here, this middle portion here is the search strategy system. You'll see all of these rectangles. I'll walk you through one in a little bit more detail in a second. But this is an example from one of our resources, ToxoDB, where I'm looking for genes that are potential cyst stage transporters. And I used many different types of data sets or data types to come up with finally with a list of genes that I may want to go back and test in the lab. What I want to point out here is that this choo-choo train of steps allows you to integrate data with each other and diverse data types. So from analyses like transmembrane signal or signal peptide domain searches to variation data from population sequencing data sets to various transcriptomic and proteomics data set, the whole genome CRISPR screen to high throughput localization from spatial proteomics data sets and so forth. So you can hopefully getting the point that you can integrate different data types with each other. And the key thing here is that you are actually leveraging data that typically would just sit in a repository somewhere or in a supplemental file in a publication, probably never touched by anyone. People would go to the paper and just look at the final conclusions of the author, whereas here you actually can go in and ask your own questions. And importantly, we really focus on the user. So our end client, which is the user, the scientist in the lab. And we try and think like a scientist. Well, what are the things that you would want to come out of a database? And we try and break down how you would go and approach the resource by asking your breaking down the question you would ask using various questions. For example, first of all, what is your main question? What is your overall strategy? Can you break it down into smaller components? And what kind of data or analyses can be used to answer these questions? So let me take you first. Next slide here. Sorry. So I'm looking at TriTripDB, again, one of our resources. All of our resources, they're basically filters of the main VEuPathDB resource, but this is something our community has requested. They wanted sort of their domain organisms housed in a single place, filtered in a single place to make it easy for them to access them. But all of them are built on the same infrastructure. So anything you try in TriTripDB, you can do in PlasmoDB or HostDB or VEuPathDB itself. We have multiple specialized searches on the left here, which you can access and open up and find searches that you're interested in. And in this case, I'm going to walk you through a quick strategy asking if I can define possible vaccine candidates in Leishmania donovani, so one of the Leishmania species that causes visceral or cutaneous leishmaniasis, a terrible disease. And so one would want to potentially develop new vaccines for this. And so these are the breakdown of these questions. So what would be the features of the candidate? When is it expressed? What life cycle stage? Where is the protein localized? Does it contain a large number of polymorphisms? Does it have orthologs or homologs in mammals? Is it essential for virulence or pathogenicity? And so walking through this, you can start building a strategy. And so you can start asking about the localization of the protein. And again, all of our searches have their own specific pages where you can go in and twiddle the knobs basically and tailor the results based on your knowledge as a scientist. And so you can go in and define the genes that have a potential signal peptide. You can then click on add step and then access all the additional searches. And in this case, I'm going to combine this search with genes that have potential transmembrane domains. And then you run this and you start building your in silico experiment, basically. And you can keep going. So I can ask for genes that are expressed in the mastigotes, specifically in the mammalian stage of the parasite, based on proteomics and RNA-seq data sets. Look for things that have a low level of polymorphisms, Dunstan-Arnold polymorphisms, so that are under pressure for change. Look for ones that are potentially essential based on orthology with another kinetoplastid, so Trypanosoma brucei. And the point here is that we have a lot of tools that allow you to leverage data across species, so you're not limited to the organism you're working on. And finally, looking for things that do not have homologs in mammals, so you're asking more of an evolutionary question here. Ultimately, you want to look at the results, which are all down here. You can explore them further. Each one of these is a gene, and you can look at their name and what they do, but you can analyze these further by looking at where they're localized geographically on the genome. And also you can analyze them using gene ontology, for example, right there on your search page, and determine whether you have some enrichment in functions. And ultimately, you can share these strategies with your colleagues in different ways. In this case, I'm showing you one where you can share it with this link, and I'll put the link right now in the chat. So anybody should be able to click on the link that I just shared in the chat, and this will open up the same strategy that we're looking up here, or you can scan this QR code if you want. And then you can actually play around with it. You can modify the steps, but this will be in your own workspace. Okay, so this was the search strategy system. Let's walk through. So each one of these genes that I showed you here has its own gene page, which is basically an encyclopedic view of all the knowledge we have about this gene. And so here I'm showing an example from VectorBase, looking at an Anopheles gambiae. So this is the vector that transmits things like malaria, so Plasmodium, which causes malaria. And so here's a gene for an odorant binding protein 27. Doesn't really matter which protein it is, but the point is that you can scroll down and you can explore different aspects of this gene page. The point here is that there's a bewildering amount of data that is accessible to any user who goes to the gene page, and allows you to explore the data from transcriptomic data to all the expression. These are all the different experiments that are available. So again, it's a lot of stuff, but the point is to put the data right there at your fingertips, so you can ask your own questions and come up with your own conclusions. We also are trying to stay current and at the cutting edge, and so we integrate new data types. So for example, here's showing you integration of single cell transcriptomic data. This is an example from PyroPlasmaDB for Babesia bigemini, which is a bloodborne disease transmitted by ticks, showing you how you can explore the single cell data. So each one of these represents a cell, and you're looking at where this particular gene is expressed. You can also delve into this further we have our the Cell by Gene app installed internally, so people can access it and play around with the metadata provided by the user, by the data provider. So you can look at the UMAP plots, for example, and explore the cells and the expression pattern. We integrate AlphaFold data, for example. This has been hot in the field, and so wherever there is data available, we integrate this right there in the gene page. So as you're analyzing, you can go in and explore the AlphaFold pattern, and then obviously link out to all of these resources so you can learn more about these structures. You can browse the data in a genome browser. So any data that can be mapped to a genome can be browsed, and again, showing you multiple different tracks here. Again, these are just to point out that the data is there, and there are ways for you to explore it. We also develop and provide new analyses. For example, this is showing you the evidence for introns based on all the RNA sequence data we have. This is probably the only place that has this, is VEuPathDB. This is obviously important for genomes that have exons and introns, because it allows you to go in and start asking, well, is this gene model accurate? Do I believe it based on the data that's available? And you can go in and actually come up with your own conclusions. Of course, you may want to analyze your own data. And so we've integrated our own Galaxy instance. If you're not familiar with Galaxy, I would encourage you to go and explore the usegalaxy.org resource. They have a lot of tutorials there. It's an incredible resource where somebody who's a novice to the world of bioinformatics or scripting at least, or using Unix commands, you can come in here and you can use the tools that are available in a user interface with drop-down menus. and you can do things like use some of the workflows that we have available for you for, let's say, analyzing RNA-seq data, and then get your results. The cool thing is that we've integrated tools that allow you to take now results from your analysis and send them back to VEuPathDB so you can integrate them into your in silico experiments. And so, for example, here is showing you taking back some of the transcript per million TPM values from this was a Plasmodium experiment that I tried out. I brought these from the Sequence Read Archive for male and female gametocytes in the sexual stages of Plasmodium. I took this data, send it back to PlasmoDB, and then I can go to my data sets, and then right there, we'll have your own data set page. This is private, obviously, because I'm sending this privately to my own workspace. And then what's really cool is that you can start asking questions about this data. So you remember we have all of these specific or specialized searches for all of our data. So this is the specialized search we have for RNA sequence data. This search now becomes available for your own data, and then you can start saying, well, show me the genes that are upregulated in male gametocytes compared to female gametocytes by at least twofold. We have some help things here on the left that show you what you're doing. And then you get your results in a search strategy, just like we did before. And you can stop and be happy with this, or you can analyze it to look for gene ontology enrichment, or you can add additional steps to ask other questions about this data. You can view bespoke graphs that are generated based on this data within the gene page or in the results page, or you can analyze it and look at it in the genome browser. So here comparing males and females where you have increased expression for these two particular genes that may be of interest. And finally, the final tool I want to show you is that we have ways for the users to contribute back to the community. So we like to use or crowdsource people's knowledge. Including ways for people to add comments on genes, so knowledge about the genes, and those appear immediately. They're not curated. There are about 40,000 user comments on gene pages in VEuPathDB that many of them are super useful. We also allow people to contribute their own annotation using a tool called Apollo, which is built on the genome browser system. And this is just quickly showing an example from Aspergillus nigulans, which is a fungus, and showing you here a gene, a missing gene that has RNA-seq evidence, and then you can go in and build a gene model, and then the gene model within overnight gets dumped and shows up in this track called the community annotation track. Okay, so getting help, we have a lot of help on the resource. So under the help menu, you can learn how to use VEuPathDB up here. This includes many different ways to interact and learn how to use the resource, including an FAQ. We have virtual events. We're happy to be hosted by your lab virtually and give you a personal whirlwind tour of the resource. We have many webinars and also workshops. In fact, right now we're advertising 2 workshops. The applications are open, both one for VEuPathDB in general and another one for fungal resources. I would encourage you to go to the VEuPathDB homepage and you'll see links there for these resources. All of our learning materials are made available. If we have any recordings, they're made available on our resource. So you can either take the workshops while they're happening, or you can take them asynchronously. So we try and make them as useful as possible for that. And finally, you can contact us. We love to hear from you. Asking us questions and letting us know about data you have is something we love to hear about, and we love to help you. Again, as I mentioned, we have a lot of useful functionalities. Impossible for any one person to know all of these functionalities. So it's always important to go to the help menus and learn more about them, but also send us a question if you have any questions. I want to quickly shout out that we have our recent update paper in nucleic acid research database issue. I will put this link in the chat as well once I'm done so people can access it. And this will give you also a quick tour of what we have in our resources. I will end by saying that we have this model of sharing data has been very successful, and other resources have adopted this similar philosophy as VEuPathDB. These are 2 sister resources, clinepdb.org, which supports open access to epidemiologic data sets or studies. It's really cool, and there's some really cool exploratory tools there, which I would encourage you to, if you're interested in clinical epidemiologic data sets, to go in and explore those. The second resource is MicrobiomeDB, which integrates microbiome data along with all the metadata and various ways for you to analyze the data including integration of data straight from Nefeli, which is another cool resource coming out of NCBI that allows you to analyze microbiome data and then integrate the biome files into MicrobiomeDB so you can do your own analysis. So I will end here, Emily, and I will put the link to the paper in the chat. Hopefully I didn't take too much time. Thank you very much.
Oh, that's great. And love that there's some opportunities for folks to learn directly with you over the next period, next couple months. So now is a time for question and answer, and you can use Slido to ask your questions, or you can use the chat. And Moni, thank you as well for sharing your links here, because I think people are going to want to know about these resources that we have just talked through. So there's the Slido link for everybody. You can ask your question there. You can raise your hand if you'd like to participate live. And I see I think we have just an initial question in here. I think it says, you mentioned LLMs. Where do you see the limits to how you can use those tools? I think that came during Monica's talk. So maybe could you talk about the limits that you see in LLM and where that supports and also isn't a tool for your advancing your work?
Thank you. That is a great question, right? For ourselves, we have been trying to figure out what are the best ways to leverage the new technology. And so for the time being, limiting ourselves to text-based generative AI, in adapting it and implementing it across Monarch. And the limit so far is the accuracy of responses that we can receive. And I will actually call on one of my collaborators here, because he will be able to share a lot of insight from the inner wirings. But there is some of the limitation is, being able to measure the accuracy with which you are retrieving these results in what kind of added value it has. This is very experimental, and we're still trying to figure out what are the best ways to evaluate the results that we're getting and to provenance correctly all of these resources. Sean O'Neill, Dr. Sean O'Neill is here. And Sean, would you like to share some additional thoughts on some of the limitations that you see? And I'm inviting him because he's one of the developers of our resources. He developed an OpenAI plugin for the Monarch Knowledge Graph using our API.
I can try. That's a broad question. Limitations of LLMs. They have many limitations. Cost and accessibility being one of them. But I would say the biggest limitation right now that we know about is, of course, that, sort of unaided, they are prone to making things up. And so, it really is on us to provide them with accurate and broadly representative information that they could use to give better results of things. And I think where we are right now is in figuring out the best ways to do that. I would frame an LLM as like a competent undergrad who maybe thinks they know more than they do, and the better we can set them up for success by providing accurate and good information, the better results we're going to get out of it. And yeah,
absolutely right. Thank you, Sean. Yeah, to be able to best inform with resources that are accurate, so that the LLM itself is best informed, is also incredibly critical. Thank you, Sean.
I can add to that as well, maybe from our end,
Yes.
if I may. So yeah, I mean, I totally agree, obviously, with everything that was said. I think one thing that we've been exploring, obviously, because we've been for many years integrating data in sort of a standard way, describing all the samples in sort of, again, a standard way. We now have a wealth of well-described data sets in our resources, which now can be fed into any sort of AI models. And so that's something we're obviously exploring. Another thing is, as you mentioned, the ChatGPT, that something like that would be really cool. I mean, I showed you that search strategy system, which you can go in and run your own little steps. Each one of these is really a question that you can imagine tethering together into a natural, language. I could say, I want to find me all the possible vaccines that have a transmembrane domain and phosphorylated and don't have expression at this stage, right? And we have all the underlying searches available. And so now the trick is to be able to link those together in a way that works and is efficient. So there's some exciting things I think coming down the road, but there's still a lot of work to be done, and it's not as simple as people think. Oh, let's throw some AI on there, and we're going to get these amazing results. There's still a lot of careful work that needs to be done to make sure that the results are absolutely right. Yeah.
Yeah, that's a very good point. Omar, I kind of want to, you mentioned in your presentation about user input comments and that being a field that, can you speak to how that has improved and sort of what that contributes to your tools and resources, and what you've seen as allowing that level of comment and participation in the tool itself?
Yeah, thank you. So we have, as I showed you, we have these gene pages, right? And gene pages are these encyclopedic views of a gene, right? There's a lot of information in there, but there's a lot of information that is really sort of in the brains of the researchers who are working in the lab. They know that they try to knock out a gene and it never worked, so they're never going to publish that information, right? But it's very useful information to have. And so what we did is we generated something very simple. We said, let's have a very low threshold for collecting user knowledge. And so on each gene page, there's a section there where you can just click and say, add a user comment. You have to log in because we just want to make sure that it's associated with a particular person. And then you can go in and add a comment. And you can say, oh, I tried to knock this gene out. It never worked. So I think it's essential. Or I knocked this gene out and it worked, and we have great publication. Here's a link to the PubMed ID, right? Or here are some images of the subcellular localization of this gene. And then as soon as I click on submit, that comment becomes available on the gene page, and it becomes indexed, so it's searchable. So any words in the comment become searchable. And this ends up being incredibly useful because now, as I mentioned, we have close to 40,000 user comments in the database, over the span of 20 years. And sure, some of them are maybe not that useful, or maybe now are superseded by really cool data sets that answer that question, but they're still user comments attributed to a specific person, and people can look at them and say, oh, okay, this is interesting, this is useful, or you know what, maybe I need to contact them and find out more about this, or okay, I'm going to ignore their comment, right? And so, but the nice thing is that it's available to them. One thing that we've done, though, is we've associated those user comments with what we call user comment campaigns in key meetings, where we actually provide something in return for the user comment, just to get people excited about it. And we found that, during these conferences, we would get, maybe a few hundred user comments during the conference. But then what we found out is that after the conference, it doesn't go back down to like 2 or 3 comments a week. It goes back down to like maybe 20 comments a week, right? So there's a lag period where people continue adding user comments. And right now, it's because we've been doing it for a while. It's sort of a natural thing that many people in the field, they say, oh, I better add a user comment about this because it's information I'm never going to publish, and it's useful to put on the resource.
Yeah, that's quite an interesting community engagement tool, and I think something that bridges that statement, as you said, that there's many things that researchers will learn throughout the course that might not be published, but would still be valuable to those who are doing similar work. I want to pause and see if there's any questions from the audience, and if not, continue the conversation. I meant to count to 10. I got to 8. I wanted to ask you, Monica, about making data sets interoperable, or making data more interoperable, and I think that is a really big topic, but also something that to hear your reflections on what's possible now and how fast that has moved in the recent time period, because I see that interoperability is kind of a frontier of using all of the research that is out there.
This is true. I mean, it's challenging. I've for a long time joked for a little bit that standards is not the shiniest and most attractive type of work when you ask people to think about the implementation of standards, or just the thought of standardization of the knowledge itself and the data that is going to allow us to draw these insights, people just go, hey, do I really have to? And so change moves at the speed of trust, and for us to be able to engage that community so that they have buy-in into how we have improved our standards for the purpose of, or how we improve our standards for the purpose of having better data sets, we have to get the community involved into this. And so it has allowed us to do things we weren't able to do before. So for instance, one of the resources that we have generated, which is the merged or a combined disease ontology called Mondo, has allowed us for the first time to look into data from a project that is focused on, this is an NIH-funded project that is focused on looking at the co-occurrences in comorbidities that occur in persons with Down syndrome across the lifespan. And there are these different cohorts, X01 studies that have been funded by NIH as cohorts for study. And all of this raw data that has been processed, you can get some churning and insight on like this percentage of this particular disease has happened into this population and this percentage on this other population. But there were no possibilities before for us to be able to actually recognize things that are happening across the studies. And so we've been working on for the last year in generating a data-based or a data-informed data model that allows us to make these data sets, talk with each other, and allows us to find points of cohesion. So I believe I'm rambling somewhat because an answer to the question of how fast it moves is a bit challenging. It moves with the community and it moves with the needs and the feedback that you are provided, specifically for our ontologies for the disease one. We have a community of experts, and so we tap into the domain expertise and clinical geneticists that are able to provide us with some feedback. And then we just also reach out to the community at large to get us some additional information on what we're doing correctly and when we're not doing things in the most adequate manner to the better representation of the disease. And so it depends on the trust that you build with the community, and it depends on your ability to strike this balance between wanting to throw the kitchen sink at a single standard so that you capture every possible fringe scenario, and being able to make a standard that is sufficiently nimble so that you can actually exchange information in interoperable manners. And because if you customize it too much, then you end up no longer having a standard.
Yeah, it's a balance, but I really appreciate both of your emphasis on developing resources that meet community needs and are really rooted in engaging the community in what they want to learn or what they want from this type of resource. So I want to commend you on that. I will do another 10-second count to see if there's any other questions from the audience. And if not, we'll wrap things up, and I have a few announcements related to DataWorks.
Mine wasn't a question. It was just an appreciation message to many of the resources that Omar is deploying, because brings me back to time of me working with the Apollo and the JBrowse teams. And so I was just happy to see all of those continuing to be deployed.
It's great. I mean, we love to use other people's tools. So
absolutely, we're
there's
research
no reason. Exactly, and no reason to reinvent the wheel, right? So if there's a nice tool out there, our goal is to make sure it's integrated and that people can actually use it, basically.
Yeah, I think that is really the opportunity here, is how we can leverage the tools that exist to do even more in those fields. And I really appreciate that folks can learn from what you're doing when they think about both their individual work and potentially other tools like these. So to wrap things up today, we just have a few announcements from the DataWorks program. I really want to say thank you to our speakers for sharing their experiences and really highlighting the role that integrating data sets and developing tools to access those can have in scientific discovery. So I believe we've got the screen here, and I want to go over a few opportunities from the DataWorks program The main one is that we have a help desk here, and the help desk is on another side of data management, which is planning for your research and developing a data management plan that is in alignment with the requirements of NIH funders. So this is a resource that is just launched and will help you go step by step through what you need to know as you're planning your research. And as I said, another part of the world here, but I think a really valuable resource to help you set your data management practices up right from the start and be able to develop high quality research practices and data collection and management. So it talks through the policy, gives you templates for how to create a data plan, making decisions about data storage, repository finders, and so much more. So we will be sending out the link there, but it's live and a resource that's free and available for this community. We'll have another Salon on December 13th. We'll be talking about how to create those data management plans using this tool